Welcome back, everyone. So in the last lecture, I was talking about muscles, and I was talking about skeletal muscle and how we excite skeletal muscle and how neurons have to innervate skeletal muscle. Now we're going to talk about the actual muscle contraction of skeletal muscle. We're going to start at that neuron and explain exactly what happens, the different steps that go on to cause muscles to contract. And so while I'm doing this, think about every single muscle in your body and how often you use them. And this all has to happen just for that muscle to move. So the first thing that's going to happen is here you have that axon end bulb. So there's this axon up here that is receiving a signal. That action potential, which is the signal, is running all along the axon, and it's running to those end bulbs. Once it hits the end bulb, it activates calcium-gated channels. Calcium is going to rush in, and calcium has to bind to these little vesicles. The vesicles that have bound calcium are then going to bind to the plasma membrane of the axon, and they're going to release their neurotransmitter, which in this case is acetylcholine, into the synaptic cleft via exocytosis. So the acetylcholine molecules then are going to bind to the receptors on the sarcolemma. This activates the receptors on the sarcolemma to open, allowing sodium to rush in, and this stimulates an action potential in the skeletal muscle. So now we have this action potential moving along the skeletal muscle, sarcolemma. It's going to move along the sarcolemma, and it's going to move down the T-tubules. And so as it moves, more sodium-gated channels are opening, flooding the muscle fiber with sodium, which causes nearby sodium-gated channels to open, which causes the action potential to move all the way along that T-tubule. And once it hits the T-tubules, it activates calcium-gated channels in the sarcoplasmic reticulum. So calcium, which is bound in the sarcoplasmic reticulum, is then released into the sarcoplasm of the muscle fiber. And I talked about the regulatory proteins, troponin and tropomyosin. So their function is to allow or is to monitor the muscle contraction. They regulate that muscle contraction. Tropomyosin sits right over the actin binding or the myosin binding sites on actin, and troponin sits on top of the tropomyosin. When calcium binds to the troponin, then troponin and tropomyosin move off the calcium. They change configuration and they move off the actin molecule, not the calcium, I'm sorry. They move off the actin molecule binding sites, and this allows myosin to be able to bind to the myosin binding sites. So this leads us to the actual muscle contraction. So now we have these open actin binding sites, or myosin binding sites on actin. I keep saying sorry. Activated myosin heads are going to attach to those myosin binding sites. This forms a cross bridge, and then the myosin heads pull the actin towards that M line of the sarcomere. So remember, the M line is the very middle of that sarcomere, and so myosin is pulling the actin towards the M line. Actin is moving towards the M line. We're actually shrinking the A band, or we're shrinking, the H zone, the region where there's only myosin, and we're increasing the size of the A band. Once we move towards the M line, myosin then is going to release the ADP and the phosphate that is bound to it, and now we have that ATP binding site open. So at this point, if there's no ATP, the myosin is going to just sit there. It can't do anything. It can only unbind if more ATP attaches to the myosin binding site. Once it attaches, then we end up having the myosin is able to unattach from the actin molecule, and the ATP can then be hydrolyzed to the ADP and the phosphate, and the myosin head will be activated again and able to bind to another myosin binding site on actin if calcium is still in the system. If not, then it'll just sit there waiting for the next time it can attach. So this leads me to the sliding filament theory of the muscle contraction. So here we have the Z disc. Here we have the actin molecules with the troponin and tropomyosin covering. Here we have the myosin. So when troponin and tropomyosin move off the actin, myosin can then attach and pull the actin towards that M line. This shortens the actual muscle length because you're contracting the muscle fiber. And then we can relax it. What we're not doing is we're not shortening the muscle fibers. Or we're not shortening the myofilaments. So if you look, the myofilaments never change in size. If you measure right here, they're the same size on both sides. And if you measure this one here and this one here, they're the same size. The difference is we've shrunk that H zone to nothing. So there's like no H zone or very minimal amounts of H zone where there's only myosin left. And that's what we're doing. That A band, which is where we have both myosin and actin overlapping or thick and thin filaments, becomes the majority of the entire structure. So on your exam, I typically do ask a question about the muscle contraction. So you do want to know the different steps in that muscle contraction and how they occur and why they occur. Also, I want you to think about this. So if you have a pen and paper, you might want to write this down. Why do people, when they die, become stiff during rigor mortis? So if you're not sure, and you probably aren't just from listening to what I've said, do a little bit of research to figure this out. So I've given you the answer, but I didn't give you the answer straight up. I told you what's going on, and so now take the information I gave you about the muscle contraction and try to figure out what's going on, okay? And now we're going to get into the next section, which is how muscle fibers get their ATP. We need ATP for muscle contraction. We need a lot of ATP. So muscles have different ways of producing ATP. They have mitochondria to produce ATP through aerobic respiration, but not all muscles use aerobic respiration or the normal mechanism for producing a lot of energy. Some muscles use other mechanisms to produce the majority of their energy. So in general, we have 3 different types of muscle fibers, and any given muscle will have a mixture of all 3 types of muscle fibers within that muscle, but there's going to typically be one type of muscle fiber that's going to be the most common. So there are 3 ways that we produce energy. The first is through taking ATP and just breaking it down immediately. So we have ATP that's just in our cells, and that stored ATP can then break down and produce a small amount of energy. We also have a high energy molecule called creatine phosphate. Creatine phosphate sits in our cells, and we can take the phosphate from the creatine phosphate, and we can add it to an ADP to produce ATP. So when we have energy stored, or when we have a lot of ATP built up in our cells, creatine here is going to, if we have a lot of energy that we're not using, creatine will take the active phosphate away from the ATP, producing ADP, which can be stored a lot easier in our cells, and producing that high energy creatine phosphate, which is also able to be stored in our cells. Then when we need that extra boost of energy, creatine phosphate then donates its phosphate back to the ADP, producing that ATP. So this only gives us a small amount of energy. This is actually going to be approximately 15 to 25 seconds worth of energy. So super small amount, but enough to do like a very fast 50-meter dash. So think about that energy that you have when you see a child running into the road and there's a car coming and you have to catch that child. That's the energy that you're using, that immediate stored energy. Then for longer bouts, maybe, and I'm making up the amounts, but you know, for an additional 45 or so seconds, so I'm the length of I'm thinking of running, okay? So the first 15 seconds, I don't know, 15 seconds is about a 50-meter dash. I don't really know if I'm correct or not. Then for another 45 seconds, we're going to receive a little bit of energy through an anaerobic process called fermentation. So we ferment, and when we ferment, we produce a substance called lactic acid. Lactic acid can then build up in our blood, and it can cause all kinds of problems, including muscle fatigue, which you've probably felt if you've ever worked out in the gym for a long period of time. This process actually is the first step in another process called aerobic respiration. So we will talk about aerobic respiration in like chapter 26, I believe, when we talk about metabolism and digestion. And you probably have heard of aerobic respiration in your Gen Bio class. So the first step of aerobic respiration is this glycolytic pathway. So glycolysis is an anaerobic process, and we take the end product, pyruvate, in the absence of oxygen. We are then going to convert the pyruvate into lactic acid so that we can reproduce another molecule called NAD that can then be help to break down another glucose molecule. So we end up producing only a small amount of energy, a net gain of 2 ATP, but it's enough to keep us going for an additional about 45 seconds. It takes us about that long for aerobic respiration to really kick in, producing a lot of ATP. So the majority of the ATP we produce is through aerobic respiration. And in this case, we're using oxygen, which is why it's called aerobic. And we're producing a lot of ATP. This here, glycolysis, gives us a net gain of 2 ATP. Up here, we're only getting that stored ATP that we had in our cells initially. And then down here, we're going to produce, for every single glucose molecule, we're going to produce about 36 ATP molecules. So very energy efficient compared to these to fermentation. This is going to keep us moving for, you know, an entire like mile run, if you think of it that way, okay? So 50-meter dash, maybe 100-meter dash, or a quarter mile. That depends on how fast you run. I mean, I'm just making this up. And then this would be the remaining part of that mile, okay? So what happens when we produce energy, or when we produce energy and we use that energy? We're going to, so the second law of thermodynamics dictates that if you produce or if you take energy and change it from one form to another, you lose energy as heat. So we're definitely going to produce a lot of heat, which is going on here. You see this guy sweating to try and release that heat. But we're also going to be in oxygen debt. Oxygen debt means that our body needs more oxygen. So it's at the end of exercise when we are trying to restore the oxygen that has been depleted from our cells. So skeletal muscle has a special molecule called myoglobin. We've talked a little bit about hemoglobin in class, specifically in chapter 2 when we talked about the tertiary or the quaternary structure of proteins. I mentioned hemoglobin is the best example of a quaternary protein. Hemoglobin binds oxygen. Myoglobin binds oxygen in muscle fibers. And so the oxygen that our cells are, or that our muscle is using comes from both hemoglobin, which is being delivered. So hemoglobin delivers oxygen to our cells, and myoglobin, which stores oxygen in our muscle fibers. And so we have to replace that. That's part of that oxygen debt. We're replacing oxygen back in our system that we have used. We're going to have to replenish glycogen stores. So we've used a lot of glucose. When you exercise, you're using a lot of glucose, and you need to replenish that glucose back. And so we take extra glucose and we build up glycogen stores. We have to produce more creatine phosphate for the phosphagen system. So that phosphagen system is this system up here. I didn't name it. Phosphagen system is this first system, anaerobic mechanism to produce ATP very rapidly versus this anaerobic mechanism here that's a little bit longer called fermentation. So we're going to replenish ATP stores, replenish the creatine phosphates. So all the creatine phosphates have been oxidized and have lost their phosphates. That's what I want to say, dephosphorylated. And so we need to add phosphates back. We need to convert any lactic acid back in the glucose into, or in the liver, into glucose, so that we get rid of that toxic material that could potentially harm our cells. Uh-oh. Hello. Okay, why is this not working now? Hold on, sorry.